Welcome to another biology video.
Cellular membranes and the fluid mosaic model.
So for fluid mosaic model, we're going to be analyzing the components of a cellular membrane and try to figure out why the fluid mosaic model is our best current model to understand the skin around a cell.
So why should we care about membranes? It's because membranes are things that surround just about everything inside the cell as well as the outer covering of the cell.
Every organelle, every specialized compartment within your cell allows for an internal environment differently than what's outside of it because of cellular membranes.
Yeah, it's surrounding the nucleus, the mitochondria, vacuoles, chloroplasts, and then the whole outside of the cell membrane, is obviously made up of the same material, cellular membranes.
If it weren't for cellular membranes, we'd be large amoebic structures incapable of standing up.
That looks kind of like, never mind.
Yeah.
So in your journal, we want you to record a couple of things. First is a timeline. And through this presentation, you're going to see 6 dates and 6 specific years of where certain things happened in our understanding of cellular membranes. We also want you to include 6 descriptions for each of those dates about what happened around those times. On your other page, we want you to include some labeled diagrams. We're going to show you what a phospholipid bilayer is. So having a label diagram of that, a model of a cellular membrane called the sandwich model, which is an old model, and our current model, which is called the fluid mosaic model.
So in your journal, we should be able to find these pictures and this timeline.
Okay, let's go. So what makes cellular membranes so functional? Well, one thing is because they are selectively permeable. So permeable means to allow things to pass through. But selectively permeable means that it's going to allow some things to pass through, like these little particles that Mr. Balsiger is drawing. Little particles can weave their way through the membrane. Big particles cannot move through the membrane.
Think of a net. The meshing of the net filters some things in and some things out.
That's right. Another thing about makes them so functional is that they are extremely flexible. And this allows for them to bend and to fold. And in those bends and folds, allows them to create a lot of surface area. I mean, we think about cells as being like these blobs that sort of sit there, but they're really dynamic. They're moving and flowing and ebbing and communicating with other cells adjacent to it. And so having a cell that's just like this round circle isn't really realistic. If we look at the membrane more closely, you'll see that it's all wavy and bendy, and so it has more surface to contact with its outside environment. And that's good. It means more chemical reactions can happen.
Think about your chicken egg. When we dissolved the shell off your chicken egg using vinegar, the egg was flexible on the inside, not on the inside, but the outside of the egg. The cell membrane that was left after we dissolved the shell was flexible.
All right. So here's the first date to put on your timeline. And this is 100 years ago in the 19-teens. Different scientists did chemical analyses of the membranes, and they found they were made up of 2 different types of molecules. A phospholipid, which is a lipid, a special kind of lipid, and proteins. So let's look at more specifically at those phospholipids because they have some interesting properties. Here's 3 different drawings, all sort of showing the same thing. Just a model of what a phospholipid looks like. But it looks like the one on the right there looks like a beach ball with 2 little tails hanging down. The beach ball is the hydrophilic region. And so hydro, like water, and philic means loving. So that's the side that actually likes to point towards water. And there's a whole other, the tails are hydrophobic, meaning they're water fearing. So they point away from water.
So let's draw a picture as an example. Here's water. Here's a phospholipid. The phospho part, the head, loves water. It's like a magnetic attraction. They love each other. But the legs, they hate water. Imagine putting oil in water. Water places the oil into a molecular slum. It pushes it into a blob and doesn't allow it to dissolve. That's because they repel each other. So if you have a phospholipid next to water, the tails are going to be pushing away from the water's surface because water repels the legs.
And the head's going to be pointing towards the water.
The head likes the water.
So this was a problem.
Why?
Well, I mean, if cells are always living in a very watery environment, how could they be filled with membranes that are partly hydrophobic? Like, how would they arrange themselves?
In other words, you are essentially a large sack of water made up of cells. If every cellular membrane is made up something that's hydrophobic, it doesn't make much sense.
So here's the solution in the 20s.
This is next on your timeline.
Next on your timeline.
1920.
Is two scientists hypothesized that maybe the membranes are made up of a phospholipid bilayer, bi meaning two. So two molecules thick of phospholipids, and they're stacked one on top of the other, like in this picture.
So here I want to begin an analogy that we're going to continue throughout the rest of the video. First, I want you to imagine that you have a pool, and in this pool you put beach balls. So many beach balls that the beach balls become the surface of the pool. So as you're looking down at the surface of the pool, you have all these beach balls floating around. The beach balls will represent the phospho part, the part that likes water. They're the surface of your pool. The inside of your pool, the second layer, is made up of these lipids. Remember, these are fatty acids being repelled by water. So the inside of your pool, the water of your pool, are these lipid tails.
It's kind of oily.
So if you were to dive through these beach balls, you would pass through an outer layer of beach balls into an oily layer in the center, and then you'd fall out. The bottom of your pool can't be a floor. It's actually another barrier of beach balls, and you end up in another environment. That's the job of the cell membrane. It separates the cell's insides from the outsides. So what passes through it's like passing your magic pool with beach balls, oily water, and beach balls as a floor would put you into a new place, like Narnia.
And so, yeah, so this sort of solves the problem. This does solve the problem because now we have a watery environment on the inside and watery environment on the outside, and the hydrophilic parts, the beach balls, are pointing toward the watery area, and the hydrophobic parts are away from the water. Brilliant. Problem solved. But now we have another problem. We know there's the proteins. Remember from earlier in the slide, we knew that they were made up of phospholipids and proteins. So where are the proteins?
In 1935, two scientists proposed the idea of a sandwich model.
Sandwich.
Where the bread of the sandwich is going to be a protein layer. Think about a sundae with a hard shell coating of chocolate on it. And the filling of the sandwich now is the phospholipids. Check it out. There's a real picture.
Here's a better picture. That was a nice picture, though. So the green little thing is supposed to be this protein coating on the outside. It's on the outside as well as the inside, sandwiching
the bread.
All that yummy stuff in there, the lipid bilayer.
The phospholipid bilayer is the filling of your sandwich.
Now, what's interesting is that this model was accepted for a long 30 years.
30 years.
That's like 2 of your lifetimes. This is the way people were taught what a membrane was like. Widely accepted.
Twice as old as you.
Until the 50s. So here's your next part of your timeline. In the 50s, there's a new technology called the electron microscope, which is this really different type of microscope that shoots electrons at a piece of material and it bounces back and we can detect that. So it's different than a light microscope like what you've used in class.
The light microscopes might go 4,000 times magnification, whereas scanning electron microscopes, I think they might be able to go 400,000 times.
So we could really look closely at these membranes. And what they found was this triple layer staining pattern that you'll see on the next slide. Now, scientists assumed that this was confirming what they thought originally, that the 2 dark lines are the protein layer, and that little whitish line right through the middle, that's the lipid bilayer.
So they patted themselves on the back. They said, congratulations, we've been right all along. Of course, they wanted to be right.
Yeah, see? Look at that. We were right. Look how smart we are. However,
in the 1960s,
Next thing on your timeline.
They discovered that the proteins are also, most of them, hydrophobic.
So they don't want to be on the outside near the water. They want to get tucked in by the tails of the fatty acids.
So now the sandwich model doesn't work because the sandwich put the proteins
on the outside.
On the outside.
So what the scientists assumed was wrong, and instead the triple layer stain
was actually
2 dark layers are the hydrophobic regions of the lipid bilayer, and the inner region is the hydrophobic part of lipid bilayer.
In other words, those dark regions are what we were making our analogy, the beach balls floating on top and bottom of your pool, and the light region was where the lipids were in the middle.
So then, where are the proteins? The next problem. If the proteins can't be in contact with the water environment and they can't be on the outside, then where are they? So in 1972, Singer and Nicholson, 2 other scientists, proposed this idea of the fluid mosaic model. And we're going to talk about this. This should be your last entry in your timeline.
Add it now.
And what they propose is that the proteins aren't on the outside, like the bread for the sandwich model. And instead, they are interspersed. They are just kind of passing their way through the entire membrane.
Here we have a picture.
That looks a lot better.
So draw this as your last diagram. You should have a diagram of the phospholipid bilayer, the sandwich model, and now the fluid mosaic model.
So these proteins act like doorways. They are where the cell can put out energy to then transfer a large molecule into the cell if it wants to, and bypass the phospholipid bilayer. And so that's their function. But it's much more complex than that because it's not just about the proteins. We now know that there's other things that are a part of this cellular membrane as well. So let's break apart the name of this model, the fluid mosaic model. So first, why the term fluid? As it says here, membrane is fluid, not solid, because fatty acids in the phospholipid molecules are unsaturated.
And what that means is molecules can slide past one another. Take our beach ball model. You could actually move those beach balls around. They can pass by one another. Also, we have to modify our beach ball model now. We have to add large floating devices like big dinosaurs and other things you'd find in pools that would be floating deeper in the pool. They're bigger than the beach balls, and they can be pushed around inside of the beach balls as well.
So this makes it so the membrane is it's moving and can change shape.
A huge difference here is that I think in the beginning they thought of membranes as being smooth and all made up of the same thing, and now membranes are bumpy and hairy and they look different.
The second concept is mosaic for the fluid mosaic model, meaning that mosaic means it's made up of many parts. An artist can make a mosaic, which is a really cool way of designing something. It's just like a really complex way of building a picture. So it's made up of many different parts. We have the proteins that are interspersed throughout the membrane. We also have things called cholesterol. Cholesterol is found in animal cells, and basically what they do is kind of put some space in between the phospholipid molecules and even help make it be more fluid when it's in warmer temperatures and less fluid when it's in cooler temperatures.
So as an ending picture of the fluid mosaic model, we want you now to think of the surface of your cells as this flexible, flowable surface that has all these little things hanging off of it. It's a mosaic.
There's also these long carbohydrate chains that are hanging off, and those help the cell to communicate with its outside environment. So like for it to be able to communicate with other cells nearby. That's a really important function in regards to your immune system.
Your immune system is trained so that it knows which cells are your own by looking at the surface of your cells, by looking at the stuff sticking out of your cell membrane. So if a cell doesn't have the right kind of stuff sticking out of the membrane, it'll attack it and think it's an invader, something trying to get you sick.
So for example, if I donated my kidney to Mr. Balsiger, his body would reject it because his body would recognize that kidney is not from his own body. He would have to get it from somebody who is much more similar genetically, who would have similar cells and similar membranes and similar carbohydrates hanging off the sides. So like a brother or sibling of some sort.
Interesting.
Very.
So thanks for tuning in, and we'll see you in class.
Bye.